In this lecture, we're going to take a closer look at the inner portion of the cerebellum, in particular the cerebellar cortex. And the cerebellar cortex, recall that is considerably thicker than the cerebral cortex, and as such, it consists of three distinctive layers. The most superficial layer is the molecular layer that we'll get into a little bit more detail here on this diagram, on this drawing here on the left. And the molecular layer consists of axons. And these are axons that transmit bioelectrical signals from granule cells, stellate cells, and basket cells. These all serve as interneurons inside this most superficial layer of the cerebellar cortex. And a major neuron in the cerebellum called Purkinje cells contain dendrites. A very extensive dendritic tree exists in the molecular layer. And recall when we talk about dendrites, we're talking about that portion of a neuron that takes in synaptic connections from other neurons. The middle layer, the thinnest layer of the cerebellar cortex, is the Purkinje cell layer, named according to the cell bodies that are contained in that middle layer. And those are the cell bodies of the Purkinje cells. And the function of this layer, as such, the Purkinje cells, will convey outputs of the cerebellar cortex. And this output moves towards the white matter that you can see here at the bottom of this drawing. And recall that white matter is that portion of nervous system tissue that is composed of axons. And the innermost layer of the deepest layer of the cerebellar cortex is the granule cell layer. And this is a cell layer that is very packed, in a very densely packed layer with granule cells. And it's estimated that there are 10 to the 10 cells that are contained within this portion of the cerebellum, making it a very dense, giving this portion a very dense, hard type of texture. And the granule cell layer forms connections with afferent terminals from a region called the cerebellar glomeruli. And the glomeruli just refers to a mass of neural connections in that granule layer. And when you pack these many neurons in one location, you're going to have numerous synaptic connections. So if we take a look at our diagram here, this drawing, here we see the most superficial layer, the molecular layer, the thinnest layer, the Purkinje cell layer, made up of the cell bodies, largely of the cell bodies of the Purkinje cell, the thickest portion of the cerebellar cortex being the innermost, deepest granule cell layer. And as indicated a little bit earlier, what we have here is the white matter, which are made up of axons. And I want to make particular note of the Purkinje cell layer, which houses the cell bodies of these Purkinje cells, and these are the output cells. So bioelectrical signals will travel in a downward manner towards the white matter. A couple of more points to make note of here that we'll address are what are called mossy fibers. And mossy fibers will send bioelectrical signals, as do what are called climbing fibers, and we'll get to those as well. But when you think about the white matter, these are not just axons that transmit bioelectrical signals downward, but these are axons that transmit bioelectrical signals upward as well. And we'll address that here in this video. This is a microscopic image of a cross section of those layers. And so here we see the molecular layer, the very thick granule layer, and the very thin Purkinje um, layer made up of those cell bodies of those Purkinje cells. And once again, the Purkinje cells are the output pathway of the cerebellum, particularly the cerebellar cortex. And the Purkinje cells receive input from what are called mossy and climbing fibers. These are neurons that transmit information up towards the surface of the cerebellum and that cortex, and will synapse onto the Purkinje cell dendrites. And the mossy and climbing fibers will also send what are called collaterals. Collaterals are sharp branches of these axons that will transmit those action potentials, those bioelectrical signals, to the deep cerebellar nuclei. So, what we see here are very extensive connections between incoming information to different parts of the cerebellum. If we take a look at the inputs to the cerebellar cortex, the mossy fiber is considered the major afferent input to the cerebellar cortex. So once again, when we talk about afferent, we're talking about incoming bioelectrical signals to the structure. And this is incoming from largely brainstem nuclei, as well as the spinal cord nuclei. So the nuclei, once again, clusters of neurons located in the brainstem and clusters of neurons located in the spinal cord via the spinocerebellar tract will bring information into the cerebellar cortex, and the mossy fibers will project to granule cells in the granule layer. And as you can see here in the picture, we'll highlight this in a moment, but the granule cell layer will then project to the molecular layer, and the molecular layer being the most superficial, at which point bifurcation occurs. So bifurcation means that there is a sharp split in these neuron axons at that level, and they form what are called parallel fibers, named according to the fact that these fibers appear to travel in a parallel manner along the surface of the cerebellum, and these will actually travel several millimeters to synapse onto the dendrites of the Purkinje cell. So although the Purkinje cell body is located in that very thin middle layer, the dendrites project up into the outermost layer and the most surface layer, and that's where it receives inputs from these parallel fibers. So one granule cell receives inputs from many mossy fibers, which are the afferent fibers, and one Purkinje cell having such an extensive dendritic tree serves to increase the probability that synaptic connections will be made. And so by increasing that probability through that extensive dendritic tree, we see that one Purkinje cell receives inputs from as much as 200,000 parallel fibers. And if we take a look at this and follow this pathway on the diagram on the left, here we have a mossy fiber, we have action potentials traveling in a superior manner where these will then branch onto the granule cells, and the granule cells will then transmit those action potentials following this synaptic connections towards the most superficial surface part of the cerebellar cortex and form parallel fibers. And it's these parallel fibers that will synapse with this very extensive dendritic tree of a single Purkinje cell. The next type of fiber is called the climbing fiber, and the climbing fiber provides excitatory input to the cerebellar cortex, and the climbing fibers originate from the brainstem region, specifically a region referred to as the inferior olivary nucleus in the medulla portion of the brainstem. And this region receives input from the cerebral cortex and the spinal cord. So projections from the cortex and spinal cord up to the brainstem will then synapse with these climbing fibers. And so in terms of function, the climbing fibers function to provide excitatory input to anywhere from 1 to 10 Purkinje cells. And so we have climbing fiber branching where it can interact with more than one Purkinje cell. Now, on the opposite side of that, each Purkinje cell set of dendrites will make contact with only one climbing fiber. And this particular synaptic connection is considered the most powerful in the nervous system, and you'll see that here on the next slide. And it's considered powerful in that one climbing fiber action potential will produce a high frequency burst of action potentials. And we can see the anatomical structure and pathway of the climbing fiber here on our diagram. So let me just pull that arrow back off. And so here we have the drawing illustrating our climbing fiber as it essentially climbs through the white matter and through the deep layer, through the Purkinje layer, and into the molecular layer where it synapses with the Purkinje cell dendrites. So if we take a look at this synaptic connection and the bioelectrical burst, on the left-hand side, here we see a typical action potential that you would find at the neuromuscular junction or along a typical axon, and the excitation that's generated between the climbing fiber and the Purkinje fiber has the same mechanism in terms of ion inflow and ion outflow. But as we move over to the left-hand side, here we see a climbing fiber. As indicated on the previous slide, one climbing fiber will synapse with just one Purkinje cell, and as you can see, wraps essentially around that one Purkinje cell and will generate these synaptic connections. And when that synaptic connection is made with climbing fiber activation, we see that the Purkinje cell output forms what are called complex spikes. So here we see very typical action potentials, so-called spikes, but when we have the climbing fiber activation as opposed to mossy fiber granule cell activation, we see a much greater degree and a much more complex degree of action potentials generated from this connection. And in terms of the role of these cells in terms of Purkinje neuron activation in terms for motor learning, here we see a rather simple experiment. And in this simple experiment, we have an expected external load handed to a person's arm. And when that happens, we see that when that load is added to a person's arm, the position of the wrist will change to accommodate that load. And when that happens, Purkinje neuron activation increases. So what we see here is a series of what are called simple spikes and occasionally complex spikes illustrated with this more complex burst of these Purkinje cells when the wrist begins to accommodate and returns back to the original position after being handed an external load, but that external load is expected. In the next experiment, we see that the same task is performed, but the load that is added to the wrist is unexpectedly heavy. So it's a heavier load that the person was not aware of. Now, what we see here in terms of Purkinje neuron activation, we see an increased frequency of these complex spikes. So if we take a look at the motor action of wrist position, we see that there's a greater excursion of the wrist and a much more unsteady degree of returning the wrist to the original position when that load was increased unexpectedly. And that corresponds with a much more complex and increased frequency burst of these so-called complex spikes, suggesting an increased level of activation from climbing fibers. And there's a decreased frequency of the simple spikes. So simple spikes are still present coming from the mossy fiber granule cell connection, but there's a much greater degree of climbing fiber involvement here. Now, as the person allows a period of time to practice with that increased load, the first item that we see here, let me just take that off the screen for a moment, is that the wrist pattern, the actual movement, resembles the very first pattern, the very first task with an expected external load. So the movement has been smooth and achieves a much greater degree of coordination. And in terms of the pattern, we can say that the person has adapted or accommodated to that increased load by practicing. And so practice will basically produce more accurate movements. But if we take a look at Purkinje cell activation, we see that there's a level of activation that closely resembles the level of activation with the expected external load. So there's a decreased frequency of complex spikes. There will still be complex spike recording from climbing fiber activation, but we don't see as much of an input from those uh, uh, neurons. And the simple spikes, upon very close inspection, appears to fall slightly below the baseline that was established here. Essentially, the adaptation occurs by way of reducing the complex spikes, which we saw in the middle task, which produced a much greater degree of jitter, if you will, a very inefficient movement to accommodate to that unexpected load. So this ends our final video on the cerebellum.